All right, we're going to go ahead and get started. Sorry for getting started a little late today. The nice thing is we don't have a lot of material to cover, but we're going to try and wrap up the aerobic respiration and general metabolism pathways that we've talked about and talk about them in the context of obesity, in the context of alcohol consumption, and in the context of oxidative stress. This is our last lecture. I think it'll be a little bit more of a breeze than our previous lectures in the past. So we left off last class. Let me actually take a step back. We left off last class tying together all the different metabolic pathways that we discussed up to this point, glycolysis, as well as gluconeogenesis in the reverse direction, the TCA cycle, as well as the electron transport chain. And so now we're going to be doing, what we're going to do is we're going to try and look at these in the context of a few different diets. And so one old standard diet that was oftentimes adopted was this low-fat diet. And so one thing to take note of is that the absence of fat does not protect one from fat accumulation. And the reason why that happens is via glycolysis, we go from glucose to acetyl-CoA. If there is an excess of glucose consumption, some of it will be stored as glycogen in the muscle, in the skeletal muscle, in the liver. But what can also happen is that if there's an excess of glucose via the beta oxidation, as well as the fatty acid breakdown pathway, acetyl-CoA, the product from glycolysis, as well as the preparation from taking pyruvate to create acetyl-CoA, it can go back up into fatty acid synthesis. And so despite consuming less fat, that doesn't, again, protect from the buildup of fat here. And basically, as I think, as a lot of us can appreciate, that typically fat accumulation can happen when there's a general excess of calories here, whether it's from either consuming an excess of carbohydrates or excess of fat here. Another dietary option here is a starvation diet. Basically, where you're consuming nothing, low carbs, low protein, low fat. And what will end up happening here is that in an effort to create nutrients here, what will end up happening is that we're going to be using oxaloacetate here for gluconeogenesis, for the production of glucose here. And so we talked before about how amino acids can be used, and via these transamination events, we can create, we can create metabolites that will then plug into either the TCA cycle or into gluconeogenesis. And so what ends up happening here is that via muscle breakdown, muscle wasting, we'll get amino acids that will then be used to derive oxaloacetate that can then plug back into gluconeogenesis to derive glucose from that. In addition to that, so that's muscle wasting. Available fat storage will be able to be used to break down to create fatty acids that'll then create either glycerol that'll then be phosphorylated and isomerized to create glycerol 6-phosphate, or fatty acids to get to acetyl-CoA. What's really important to note here is that as we all can appreciate, whenever we're in a starvation context where we have low blood glucose, you get a little haze, you get brain fog, for lack of a better phrase. That is the state in which your brain is recognizing this drop in blood glucose, and there are going to be hormones that are released in response to that. And these hormones are going to be signals that help try and mobilize these other sources of nutrients here. So one thing that's going to end up happening here is that acetyl-CoA will be able to be used to form these ketone bodies, these two carbon groups that are joined by a carbonyl. And what's key to note here is that these ketone bodies are used as energy substrates in highly metabolic organ systems. the brain is a highly glycolytic organ system. And so what ends up happening is that in a starvation context, the brain's able to use ketones because of the shortage of glucose here. And so that's going to be one other way in which we go from fat to acetyl-CoA to ketone bodies as a energy resource for the brain here. And yeah, that'll wrap that one up. One previously popular diet here is a high protein, low carb diet. One historical example of this is the Atkins diet. Atkins is probably like the 90s version or 90s name of this one was popularized. You can probably think of this now as something similar to like perhaps like a paleo diet or something akin to that. So this is effectively going to be high protein, low carb, low-ish, medium-ish on fat consumption here, but depending on the right fat sources here. What's going to end up happening here is rather than relying primarily on complex or simple carbohydrates for the source of glucose here, we're going to be relying on amino acids from the protein that's consumed to provide the oxaloacetate. Again, these amino acids that are then undergo a transamination event to go from the amino acid to a metabolite that can be used in these pathways. And in this case, it's going to be oxaloacetate that can plug into the TCA cycle. And in addition to that, we're going to be able to have the fats that are consumed, again, to be broken down to create either acetyl-CoA or glycerol that can feed into glycolysis. Typically, these high protein diets work well with a pretty intense exercise regimen because again, with the excess of amino acids that are consumed via a few of these pathways, the excess of calories that are consumed can still lead to fat accumulation here. And so typically this kind of diet, whether it's Atkins or paleo, it requires pretty careful monitoring, and that is usually combined with an exercise program. And what I forgot to say on the last slide was that usually if you return to like a pretty normal diet after that, whether it's high protein, high carb, it's usually pretty easy to regain the weight that was lost on this diet in the past. So usually the best and most effective way is a combination of some sort of lifestyle exchange that is a combination of a caloric deficit, a diet with a caloric deficit that's combined with exercise, both of which in combination can lead to lipolysis, which is the breakdown of fat. And so that the lipolysis usually occurs in combination with a decrease in fat consumption, as well as mobility that leverages the available fat as energy substrates. Because usually in combination with exercise, it's a pretty interesting metabolic change, but exercise has been shown to actually increase the mitochondria in your cells. You get more mitochondria and they become more active. And so it's a great way that fat is able to be used and metabolized when there's less fat being consumed. What's also key is low alcohol intake, because as we'll talk about in a moment, excess alcohol consumption can lead to fat accumulation. Before we jump forward, I want to pause and ask, are there any questions on the things we've talked about with a few of these diets or any of the pathways we've talked about so far? Darius and then Ashley. Yeah.
So for the starvation diet, does the muscle breakdown, I guess fatty acid breakdown, does that happen?
Yeah, typically. I mean, it's kind of yes. Typically, when a person is in a starvation state, there's you're using, because again, like if there's so many metabolic needs, whether it's And it also is driven by how active you are as well, even in that starvation state. So I know there's theories or thoughts on people doing exercise in a starvation state or fasted workouts. So typically what will end up happening is the more active you are, the more of your either fat or muscle wasting you're going to go through. And so it's not that there's going to be a shift in one versus the other. Perhaps in fasted workouts, you might be relying more on the breakdown of fats as a metabolic resource. But in general, when we talk about starvation state, it's not that there's a preference between one versus the other. It's kind of using both, trying to get as much metabolic substrates as possible. Ashton?
What did you say was the important for people off because what does that lead to?
It's a good diet, for lack of a better phrase, or if the goal is to lose weight or lower fat content, low alcohol intake is important because excess alcohol consumption leads to fat accumulation.
Is there like a delay in your metabolism? Like, does it produce certain enzymes switching from starvation to some kind of like other diet?
It doesn't necessarily have to produce certain enzymes, but usually there are hormones that are released during the starvation state that typically trigger muscle breakdown of muscle as to try and get amino acids, as well as the mobilization of fatty acids from fat storages to try and use them as another also metabolic substrate. And so there's like this central change that happens. Not that these enzymes are changing, but rather these pathways are triggered by these hormones when you enter into the starvation state, when your blood glucose drops to a certain level and stays at that low level for a while. Yeah.
So it's just like produced because there's too much acetyl-CoA.
They're derived from acetyl-CoA. There's an enzymatic reaction that uses acetyl-CoA to produce ketone bodies, and ketones are in select organs a good substrate for metabolism, for lack of a better phrase, for as an energy substrate. And so, as an example, like the brain is like a highly glycolytic, fast energy energy usage organ system, for lack of a better phrase. And so it's able to use ketones as an energy substrate or to produce energy from it. And so there's been some studies that have shown I think you might hear that either omega-6 fatty acids or fish oil is good for your mental health. And part of that's because these lipids that you're consuming can be used to create ketones that are also helpful for your brain. That's like the logic behind that.
Try to increase the production of ketones.
As I understand it, ketogenic diet is a high-fat diet. That's actually a good one. I should have included that in here. Ketogenic diet is shifts towards a high-fat diet, and the idea is that you're pushing your metabolism to try. This is as I understand it. Because I haven't included it, I haven't done a lot. I have not done a ton of background research on it, but this is from my own experience dabbling in the keto diet. Largely a high-fat diet that tries to push your body towards leveraging lipids that you're consuming, as well as lipids in your fat storages and your fat depots, and trying to use all of those lipids as an energy substrate here. And so it sounds like a great idea, but you're also going to be consuming. I felt like when I tried to do it, it felt like I was consuming an excess of lipids that made me wonder, could this also create a problem with cholesterol? Where you know, it's funny to think about this now. There's like a popular keto item called like bulletproof coffee, where you have coconut oil and a stick of butter that you have as like your breakfast coffee drink. And it is a ridiculous amount of butter that is not sustainable, I think, for any individual anywhere. So it's issues like that I think that can, like, theoretically, I think could work. Practically, I think you run into some issues. Last question.
What does that book that increased oxidative capacity of more minus one tell us about it? How does more minus one?
Yeah, so I said it really briefly. I should have kind of stayed there a little bit longer. The trying to achieve lipolysis, the two ways in which that can occur are in combination with diet and exercise. The diet part is where you have less fat, less lipids that are being consumed from your food. And in addition to that, the exercise enables increases the mitochondrial content in your cells. And what ends up happening is that increased mitochondrial content increases the oxidative capacity, which tries to leverage, because there's more mitochondria, they're going to want more substrates to use to drive these oxidative phosphorylation pathways. And so they're going to be able to metabolize fat to try and mediate that need. Yeah. All right. So to try and help illustrate some of this, we had someone in the past who did a vitamin water experiment where they were consuming vitamin water basically every day at their lunch, which is about 200 calories. And they reached a point, you can probably tell this is not me, surely based on the weight. But this person stopped drinking vitamin water for a while, and they're able to see that over time, that once they stopped drinking vitamin water, that they were able to see a pretty clear trend downwards. But there's a lot of ups and downs, which is usually the case with weight, that there's some degree of oscillation. Particularly, two of the large spikes coincided with this person going to a wedding where they really enjoyed the open bar, and we're going to actually talk about how alcohol consumption can lead to changes like that, where you see increase in weight. And so, for lack of a better phrase, we're going to talk about why alcohol can increase fat accumulation. I don't like the title. I don't want to say it can make you fat. I'll say it can lead to fat accumulation. So we talked about during alcohol fermentation, during the first lecture of this entire series of lectures here, that we go from acetyl-CoA to acetaldehyde to ethanol. Interestingly, when we're taking this, when you have ethanol that you're consuming in the form of drink, via alcohol dehydrogenase, you'll produce acetaldehyde, and then via acetaldehyde dehydrogenase, that'll get you to acetyl-CoA. And acetyl-CoA, again, can be taken, and we can reach acetyl-CoA via the breakdown of fats, or we can go from acetyl-CoA to the synthesis of fatty acids into lipids. And so the ability to go from ethanol to acetyl-CoA is how, when we have a buildup of acetyl-CoA, it can produce some of these lipids. And so what's key to note here is that this bullet point talks about it being a reverse action of alcohol dehydrogenase here. It's called a reverse action because typically alcohol dehydrogenase goes from acetaldehyde to ethanol in metabolism typically. And so the only reason why it's called reverse here is because we're going in the opposite direction. And so finally, these also coincide with, because this is a dehydrogenase, because this is an oxidation reaction, we're getting our reducing equivalents NADH at each of these steps as well. And so, as I mentioned a moment ago, once we get acetyl-CoA, that's actually the raw material, the actual substrate that can be used to produce fat, going from acetyl-CoA to fatty acids to triglycerides. What's important to note is that part of the reason, So not only can you get fat accumulation, but the more common appreciation of the effect of alcohol is that it can make you sick. The reason why alcohol can either make you feel sick or make you feel hungover is that the intermediate of this pathway, acetaldehyde, is not a normal product of mammalian cells. It's actually quite toxic. And so that's the key molecule that promotes the hangover effect and promotes the negative effects of alcohol. First is that it can cause mitochondrial failure, increasing generation of free radicals, and so this can lead to a lot of oxidative stress, that lead to a lot of inflammatory issues. It can also deplete our antioxidants, and we're going to talk about the important role of antioxidants and their ability to, for lack of a better phrase, deactivate or saturate up free radicals. And another key thing is that the formation of acetaldehyde, it can create the formation of adducts, which are these like foreign chemical groups on different molecules, and that can create a neoantigen. So we talked about antigens as as what our immune cells recognize, whether it's foreign or self. But what ends up happening is that these adducts, these foreign chemical groups on a molecule or on a cell or on some molecule, for lack of a better phrase, it creates a change on something. For example, let's say it is doing that on, for example, collagen. Just as an example, our body sees collagen and recognizes it as self and that it's okay. But these adducts create these foreign changes, these foreign chemical groups that'll then change collagen as the antigen and create it as a new antigen that our immune cells might now recognize as foreign and drive a negative immune response. And finally, the acetaldehyde can activate our collagen secreting fibroblasts to lay down excessive extracellular matrix protein collagen within the liver. And so you end up getting scarring or fibrosis in the liver in response to tremendous amounts of alcohol consumption over long periods of time. And so the accumulation of fat from acetyl-CoA in response to alcohol consumption, the cells dying as a response to mitochondrial failure, the immune attack from these adducts, and the synthesis of collagen are all actual hallmarks of cirrhosis. And so cirrhosis is that is kind of the end stage of acetaldehyde effects over long periods of time. Are there any questions here about alcohol consumption as it relates to fat accumulation or sickness? Yes.
Should we say that it is cirrhosis or is hallmarks of cirrhosis or what exactly?
These bullet points in the middle are all hallmarks of cirrhosis. Okay. As well as I should say the bullet points above. There's 4 things in the last bullet point. They each kind of correspond to the 4 bullet points in the middle as well. And so like all of those tie together to create these like hallmarks of like what cirrhosis is. Cirrhosis is like fibrosis or like scarring in the liver, massive cell death, really robust immune responses that are dysfunctional, for lack of a better phrase. And also you'll actually be able to see fat accumulation on both internally and on the surface of the liver as well. Yes. Yeah, so it's just that the step with alcohol dehydrogenase between ethanol and acetaldehyde, in metabolism, we're going from right to left in that box above. So typically we're going from acetaldehyde to ethanol when we're trying to break down, alcohol fermentation typically is happening in microorganisms, for example, like yeast. Like the yeast this is happening via, this is like, for example, in the production of beer and the production of champagne as well, like these reactions that drive ethanol production that also are producing carbon dioxide. This is going from right to left. Alcohol consumption in humans is going from left to right. And so that's why it's said to be a reverse reaction. Yes.
If acid so bad, why is the body making
Body is not making it. Great question.
Oh,
It's produced enzymatically in the breakdown of ethanol. We're not producing it ourselves, but rather this reaction. So, like, for example, if you decided for the next month to go completely dry and not drink any alcohol, you would not have any acetaldehyde in your body. Does that help clarify that? Okay. All right. One other diet that's actually been slightly more popular as of late compared to the other diets we've talked about is intermittent fasting. Intermittent fasting, there's a few different ways to do it. What's depicted above is 5-2, which is usually 5 normal days and 2 fasting days, where you're in a fasting state for 12 to 16 hours. You can do 8-16, where you're fasting for 18 hours every day and only eating for 6. There's a few different ways, but the key thing to note here is that glycogen stores will be depleted from the liver after about 12 to 16 hours of fasting. And so once you hit that number of 12 to 16 hours, that's typically a fasting state, for lack of a better phrase, where you're actually able to deplete your glycogen stores. And so whenever you return back to eating, you're not going to be eating so much where you're going to be, you will not be accumulating extra or surplus glucose, but rather replenishing the glycogen stores in your liver and your skeletal muscle. What ends up happening here typically is that there's going to be a degree of muscle breakdown as well as fat breakdown in this fasted state. In addition to that, it's going to trigger a mild state of ketosis where there's going to be ketone bodies that are going to be produced. That also promotes increased sensitivity to insulin so that you're able to, we talked before back, feels so long ago now, where during our diabetes lecture, where typically in response to an elevation in blood glucose, insulin will be released, and insulin will bind to insulin receptors on cells that opens up transporters for glucose to be shuttled in. And so if the connection to insulin to glucose is that insulin enables glucose to be transported into cells to be used as an energy substrate. And so even in this state of ketosis, you're actually able to have increased sensitivity to insulin so that whatever glucose is available can immediately be used. And whenever you return back to eating from your fasting state, it's typically you're just trying to replenish what's been depleted, and you won't be storing, you won't have an excess of anything that'll then be stored for fat. So our test subject, after doing their vitamin water experiment, decided to start doing a 5-2 intermittent fasting schedule where they fasted for 2 days of the week. And what they were able to see over the course of a few months was that going down to 600 calories a day in their fasted state, they were able to see a pretty clear and consistent weight loss. And so this is a pretty, there's different ways to do it, but intermittent fasting is seemingly a pretty reliable way to lose weight if you have the discipline and stamina for that. What ends up happening, I think I'm actually missing an animation here. Okay, sorry, forget that. So typically what ends up happening is during metabolism, we talked about how there can be a lot of electron leak during the electron transport chain. And those free radicals can actually lead to a fair amount of oxidative stress. And it's typically what ends up happening is that oxidative stress is this imbalance between some of the oxidizing compounds that try and get rid of these free radicals, get rid of these excess unpaired electrons that can actually create a fair amount of damage. One example of these oxidizing compounds that drives oxidative stress is reactive oxygen species. One example is superoxide that has an unpaired electron here, or peroxide. Another species are reactive nitrogen species, where there's nitric oxide with an unpaired electron. And we talked about nitric oxide a while back with second messengers, with guanylyl cyclase. But what ends up happening is that these reactive species can actually interact with each other, whereby we can actually get peroxynitrate, which is an incredibly reactive and harmful reactive species. These reactive species are typically generated from electron leak during aerobic metabolism. And those unpaired electrons, again, are highly reactive, that can create these reactive species that can drive a lot of oxidative stress. So just to revisit this slide here, we talked about how the electrons are donated by what was before our reducing equivalent is now undergoing oxidation and donating the electrons here to flavin mononucleotide, FMN right there. And so the passing on of electrons over time, whether in complex 1 or complex 3, we can have the loss of electrons. We talked about the max theoretical yield of ATP. This electron leak is what changes that max theoretical yield. And so it's primarily from electrons going from our reducing equivalent from NADH to FMN to flavin mononucleotide. And the same thing happened at complex 3, but this is the first step where electron leak can occur. And there's also typically oxygen that's going to be used for the formation of water at the end. But when there's that electron leak, these oxygen molecules can take on an unpaired electron and create the basis for our reactive species here, like peroxide or superoxide, forgive me. And what ends up happening is that these reactive species will typically modify different molecules, and they'll typically modify seemingly innocuous molecules or seemingly normal molecules that will become problematic. And so one example is the ability of superoxide to carbonylate the side group or side chain on arginine. And this, what ends up happening here is that this carbonylation on the side chain for arginine creates an aldehyde that are super reactive that can cross-link to free amines. And so it creates this what ends up happening is that these reactive species make modifications on normal molecules that then make them super reactive to interact and promote dysfunction in normal biological processes. And so another example is that they can also this oxidative stress example here can also promote reactions with lipids in our plasma membrane. And so what ends up happening is that these can actually create a reactive species on our phospholipids in our membrane that then can propagate, again, one reaction can propagate other reactions. That this reactive species or this reactive molecule can create other reactions that drive dysfunctional events. And so the ability of superoxide to affect lipids on our plasma membrane, these lipids are then modified and can then propagate other modifications that can really compromise the cell membrane. In addition to that, and you might already be seeing as we march through this that this can affect proteins, this can affect lipids. In addition to that, it can affect nucleic acids, particularly DNA, where there is going to be DNA damage events that occur as a result of these reactive oxygen species. There's going to be particular base pair changes that are going to be less, for lack of a phrase, less clean than what we've discussed in the past, where there's going to be a lot more repair machinery required to take care of these oxidative stressed, these base pair changes that are mediated by oxidative stress. So if oxidative stress is so problematic, if it can affect proteins, lipids, and DNA, surely there's a way to mitigate its effects. And there is. We typically have antioxidants that try and inhibit or prohibit the effects of oxidative stress or free radicals. And so typically these antioxidants are going to be scavenger enzymes. They're going to be these enzymes that catalyze reactions, bless you, that provide a sacrificial substrate for this unpaired electron, where typically oxidative stress, this unpaired electron will create a massive change in other molecules that drive inflammation or cell death. These antioxidants, these scavenger enzymes, will actually have a substrate that they can sacrifice to this unpaired electron, where it will, for lack of a better phrase, it will take away the effects of this free radical or make it inactive, for lack of a better phrase. And so it's primarily the sacrificial substrate on these enzymes that's key, that'll pair with that unpaired electron. One example here is glutathione. That's primarily an antioxidant, and so the sacrificial substrate for glutathione is this sulfohydryl group, the thiol group right there. And so as an example, we have oxygen that gets an unpaired electron that becomes superoxide. And typically what will end up happening is oxygen can go to hydrogen peroxide, but and that's the goal for how we want to try and inhibit this reactive oxygen species. And so we have an enzyme here called SOD, superoxide dismutase, that'll then promote superoxide to become hydrogen peroxide. And then via another enzymatic reaction, we'll create two water molecules and oxygen. But via glutathione peroxidase here, this thiol group, that's going to be the sacrificial substrate here, will enable us to, and this thiol group will enable the formation of disulfide bonds and enables us to create this inactive glutathione that's has a disulfide bond between them and two water molecules. And so it's the key thing to note here is that these scavenger enzymes are a really incredibly important way to inactivate these reactive oxygen species, and they do so by their sacrificial substrates that are able to take on the unpaired electron. So for a few of you that have proposed for your projects looking at oxidative stress, the few of you who've done that can appreciate this, but oxidative stress is a huge area of ongoing research that is incredibly hard to study and understand because there's so many different molecules that are affected that propagate down this effect, and it's hard to pinpoint down specific pathways sometimes. And so, as we talked about in the previous slide, the ability for these reactive species to target different molecules, they're able to target proteins and lipids and nucleic acids. They can themselves become a variety of different molecules that can make it hard to track where it began. This isn't quite the same as like phosphorylation events that we can actually use antibodies to target and measure. We can measure enzymes and their activity, but it can be hard to measure the actual products here from these oxidative stress reactions. And there are ways to mitigate them, as we talked about, for example, as superoxide dismutase. For each of these reactive species, there are a few different things that you can measure and look at. You can see what they affect, you can see what they become, and then what they're mitigated by or regulated by. And it's going to be different for different reactive species. And so there's a complexity here that emerges that can be really hard to track and study. But again, as you can imagine, as we've talked about how detrimental and problematic some of these molecules are and what their downstream consequences are, it is important to study. It's fairly hard to parse out what begets what and where it where it ends up. And a lot of this can be informed or mediated by specific conditions, whether it's the pH, whether it's in the presence of specific ions or other molecules here. And then there's almost a exponential complexity, because whatever they become has its own effects and things that they become and things that they're mitigated by. If you are interested in metabolism, we have a few really big names in the field that look at this. Thurl Harris is a big name in fat synthesis as it relates to metabolism. Zhen Yan is a great researcher here in the Cardiovascular Research Center that looks at metabolism and relates it back to exercise and how the two inform one another. Taking a step back outside of the university community, Alyssa Hasty at Vanderbilt is a really big name in immunometabolism, how immune cells and their own metabolism can affect inflammation and immune responses. And in BME up at Temple up in Philly, Leah Bellis, she's becoming a really big name in developing biomaterial and engineered models that tries to understand how adipocyte behavior can change in different metabolic diseases. With that being said, I want to thank you guys for a great semester. I want to apologize for getting things back late to you at times, because that's on me. But I really enjoyed this. I really enjoyed the semester. I feel like this was the first semester where I didn't feel totally inept from the first 2 times where I taught it. But yeah, I just want to say thank you guys. I still don't understand how I got a teaching award, but that's probably like a degree of like imposter syndrome on my part. But yeah, if you guys have any questions, I'll be hanging back. But I want to say thank you guys, and hope you guys have a good summer.